The overall goal of this conceptual method is to determine whether pharmacological treatments for experimental autoimmune encephalomyelitis demonstrate their central nervous system protection during the onslaught of immune cell infiltration or as a consequence of immune cell infiltration suppression.
This method can help answer key questions in the neuroimmunological field, such as how does manipulating EAE treatment times differentiate the effects on immune cell infiltration versus protection. The implications of this technique also extend toward the therapy of multiple sclerosis, since there are currently no neuroprotective treatments for this disease.
We first had the idea for this method when we discovered that treating EAE mice with an inhibitor of the System XC transporter was both neuroprotective and immunomodulatory. Though this method can be used to study drug treatments, it can also be applied to studies using genetic knockout mice subjected to EAE or other manipulations.
Begin by using sterile scissors to mince the brains and spinal cords from individual animals into small pieces. Then use one 3 ml syringe plunger per tissue to mash each sample over individual 70-micron cell strainers into individual 50 ml conical tubes while rinsing the strainers with medium. When all of the pieces have been macerated, bring the final volume in the tubes up to 50 ml with fresh medium and centrifuge the cell suspensions. Resuspend each pellet in 4 ml of 40% density gradient solution. Then carefully layer the cells slowly down the walls of individual 15 ml conical tubes on top of 2 ml.